Hello everyone, welcome back to my channel. In today's video, we will discuss about upstream processing. By the end of this video, you will clearly understand what upstream processing is, its major steps, and its importance in industrial bioprocessing. So let's begin. What is upstream processing? Upstream processing refers to all the steps involved before the actual product formation in the bioprocess. In simple words, upstream processing occurs before the product recovery, and it mainly focuses on the growth and production condition. It includes everything from microorganism selection to cultivation in a bioreactor. Basically, in upstream processing, a desired inoculum is prepared and a desired media is prepared, and then they are added into the bioreactor for cultivation of microbial cell in a controlled condition so that they can produce a desired product. Now let's look into the different step of upstream processing. The major steps are selection of microorganisms, strain improvement, media formulation, sterilization, inoculum development, fermentation, bioreactor operation. So let's discuss each of them in detail. Step 1, selection of microorganism. It is first step where a suitable microorganism or cell line is selected. It can either be bacteria, yeast, fungi, plant or animal cells. There are some parameters for this selection of organisms, like they should have high productivity, high genetic stability, and they should have ability to grow on a cheap substrate, and they should be non-pathogenic in nature. For example, E. coli strains are specifically selected for insulin production, or Saccharomyces cerevisiae are selected for the ethanol production. So the selection of microorganisms are very specific for the product formation. Now, second step is strain improvement, where microorganisms are often genetically improved for increasing their yield. The main methods which are used for strain improvement are mutation, genetic engineering, recombinant DNA technology. In this step, the specific selected microorganisms are improved. So it's a very important step for improving microorganism strain, so they can give a higher product yield and also the faster growth, which is very much important for the fermentation process. Step 3, media formulation, where a desired media is prepared for the microbial growth, and the media will support the growth of the microorganism. The culture media will provide nutrients which are required for the microbial growth and development. The main components which are used for media formulation are carbon source, nitrogen source, minerals, vitamins, and growth factors. This should be included in media so that they can support the microbial growth. Media should be cost effective and sterilized so it won't contaminate the cell culture. The step 4th, sterilization. Sterilization is essential process for preventing contamination. This mainly includes heat sterilization method, filtration, or chemical sterilization methods. These are the various different methods which are included for the preventing contamination, and they are mainly used for the sterilization process. Now the sterilization is done for the media and equipments or the air which is present, and for the bioreactor. So these things are mainly sterilized before entering in the fermentation process. If once the process starts, there is no chance for sterilizing the things. So everything, every equipment's media, bioreactor and the air supply, they are sterilized before starting of the process, so that there is no chance of contamination should occur. Step 5, inoculum development. Inoculum is the starter culture which is used to initiate the fermentation process. Inoculum is mainly developed from the stock culture of selected microorganism. Now, a inoculum should always be good, and the parameters which fit for saying that this inoculum is good or not is that the inoculum should actively growing, so it's considered as a good inoculum. The inoculum should be contaminated free, and the inoculum should also have a physiological stability. So these are the various parameters which should be checked before the inoculum development or inoculum in the inoculum development process. Now the step 6, which is fermentation or bioreactor operation, it is the most important stage of upstream processing where microorganisms are grown in a bioreactor fermenters. And during this, the parameters or the conditions are very carefully monitored. The main key parameters which are controlled in this operation is temperature, pH, oxygen, agitation, nutrient concentration. These parameters are very important and they are checked very carefully, so they can't lead to the any kind of error in the product formation. So they are checked very carefully and they are monitored very specifically in the fermentation operation. It is a process in the where the growth is occurring, the log, the lag, and the all phases are seen in the fermentation process. Now there are different type of fermentation like batch fermentation, fed batch fermentation, continuous fermentation. They are the various fermentation process where the cell growth or microbial growth or culture is taking place and the product formation is occurring. Now the upstream processing ends here and the desired product is formed. Now the desired product will be go into the downstreaming processing for the product recovery. Now so the upstream processing will be end in this step and the culture is ready for going into the downstreaming processing. Now let's look into the importance of upstream processing. So upstream processing ensures the purity and quality of microbial cells. It determines the final product yield, reduce contamination risk, improves process efficiency, and it's essential for large scale production. And one more thing, that this upstream processing is the initial phase for developing the products such as antibiotics, insulin, vaccines, and hormones. That's all about upstream processing. If you found this video helpful, like, subscribe, and comment. Thank you for watching.